Hello and welcome to Redeye. Today I have Herantis Pharma with me and CEO Antti Vuolanto. Welcome, Antti.
Thank you.
So it has been a while since we spoke last. Could you briefly start to summarize Herantis Pharma as a company and maybe your lead candidate?
Yes, so Herantis, we are a public company listed in Helsinki First North, and we have the ambition to stop the progression of Parkinson's disease. And our lead candidate, as we call it, HER096, is a small peptide that mimics the activity of the neurotrophic factor called CDNF that the company has studied earlier. And based on CDNF, we designed HER96 to capture the full activity of the neurotrophic factor that we have shown in preclinical settings. The main advantage is that with HER96 we can use subcutaneous administration. With the neurotrophic factor, it had to be administered intracranially. And now the program with HER96 has advanced very nicely over the past couple of years, and we have completed a phase 1 program. And in phase 1 we studied HER96 both in healthy volunteers but also in Parkinson's patients. And we have very strong data set demonstrating the safety of the compound, very efficient brain penetration, and then we have confirmed the dosing that we will use further in phase 2 trials, and we have also shown a biological response to HER96 treatment in Parkinson's patients, and those data demonstrate that the changes that we have observed are aligned with the proposed mechanism of action and also very centrally involved in Parkinson's pathophysiology. So we believe that we could not be in a better position to go towards a phase 2 efficacy trial that we are currently planning to start conducting.
And you presented at ADPD, I believe it was data from this phase 1b trial. What were the impressions from the audience and from people listening to you?
The most exciting part of phase 1b or phase 1 data was the biomarker data, where we showed both in CNS-derived samples but also in peripheral samples that we are affecting very broadly proteostasis, mitochondrial function, and also inflammation, exactly aligned with the mechanism of action. And as already mentioned, very centrally involved in Parkinson's pathophysiology. And based on the feedback that we have received at the ADPD and also while having discussions with specialist investors and pharma, there is a clear agreement that the data is very strong phase 1 data, and the biomarker data where we have shown similar effects on across different sample types and analytical data layers shows that as the data is very much in concordance with each other, it is definitely a clear and true finding demonstrating that we robustly address the pathology. And we are kicking off the biological process that hopefully eventually results in symptomatic improvement and disease modification. And I think the audience that we have discussed this data with, they all agree about this assessment. So it brings us in a very strong position among the compounds or companies working in the space of Parkinson's disease modification.
For example, Roche also presented the 5-year data from prasinezumab. Can you comment on that? Maybe you mentioned this a bit, maybe about kind of the competitive landscape and how you differentiate from other modalities when it comes to disease modification.
Yeah. So if we start with Roche, so prasinezumab is an antibody that is directed to alpha-synuclein aggregates to decrease the amount of alpha-synuclein aggregates within the brain. And in their phase 2 trial that trial failed in their primary endpoint. However, in extended trial, they started to see signals that are interpreted as a long-term disease modification and starting to see the first signs of symptomatic improvement. And based on those data, Roche has now decided to initiate a phase 3 trial in early stage Parkinson's patients to study prasinezumab in more details. Of course, if you compare their mechanism of action with ours, Roche's compound is very much targeted to alpha-synuclein pathology and in a way cleaning the garbage or the bad guys from the brain. If we then compare that to what we are doing, we actually are supporting the functional restoration of the neurons, very broadly addressing the underlying challenges in terms of imbalance in proteostasis and ER stress and inflammation. So there is somewhat of like overlap, but they are definitely not competing. And in longer term, you could very easily see that there might be a synergy between these different approaches. And if we take this in a way more general view on how Herantis HER96 is positioned across Parkinson's disease modification, this is exactly where we are. We address the underlying pathology in a very broad way. We have shown that in preclinical settings that we have very robust effects on proteostasis and mitochondrial function and inflammation. And now we have been able to repeat those results in patients so that we show that, yes, we have initiated those processes that we already seen in preclinical settings. And that makes us really unique, as the other compounds are more specific to certain type of pathology, typically either towards the alpha-synuclein pathology or towards certain genetic derived diseases like LRRK2 mutations or GPA, which represent only a small fraction of all the patients. So in Parkinson's disease, approximately between 5 to 10% represent a genetic background, and the rest are sporadic. And we believe that HER96 is not limited for a certain type of disease.
You mentioned there could be some synergies. Does that mean that you could have a combination therapy in some way in the long run?
Yeah, I believe that in long term, we will see gradually individual compounds entering the market, Parkinson's disease modifications. And once that happens, of course, an obvious thing is to start combining these different treatment modalities. And I believe that within the next 2 decades or so, there will be several disease-modifying compounds and their combinations used to manage Parkinson's disease. And very much similar to what we see, for example, in oncology, that there are many different kinds of treatments and they are combined to get personalized treatment for best efficacy for each patient.
And you mentioned that HER96 is